In this video, we are going to talk about DNA structure and more specifically about major and minor grooves of DNA. To fully understand this, it's important to review briefly the central dogma of molecular biology. This term describes the order in which genetic information flows. Genes contain the necessary instructions for the development and maintenance of all organisms. The central dogma has three main steps. First, in replication, DNA makes copies of itself. Later, DNA is copied into RNA, and this is called transcription. Finally, RNA encodes for proteins, and this is called translation. DNA structure is divided into four parts: primary, secondary, tertiary, and quaternary structures. The primary structure of DNA consists of a string of nucleotides that are connected together by a phosphodiester bond. Nucleotides have three important parts: a sugar molecule, a phosphate group, and a nitrogen base. The four nitrogenous bases in the DNA are adenine, thymine, cytosine, and guanine. The term secondary structure refers to the DNA double helix, which consists of the sugar phosphate backbone and the nitrogenous bases. The double helix also has hydrogen bonds, which connect the two strands together. The tertiary structure refers to the double helix arrangement in the three-dimensional space. And finally, the quaternary structure refers to the organization of nucleic acids in the context of chromatin, where DNA is wrapped around histones to form nucleosomes. In this video, we will focus on the secondary structure of the most common type of DNA, the so-called bDNA. bDNA is shaped as a double helix, which means its two antiparallel strands are wound around a common axis. The structure is stable thanks to the hydrogen bonding between complementary bases. Guanines pair with cytosines via three hydrogen bonds, whereas adenines pair with thymines via two hydrogen bonds. In the secondary structure of DNA, all the bases are on top of the other, with a hydrophobic interior and a hydrophilic exterior. The sugar phosphate backbone is hydrophilic, which means that it's attracted to water. The nitrogenous bases are hydrophobic, which means that they are repelled from water and they stack together to expel water from the DNA interior. This structure provides stability to the shape of the double helix. In the double helix, the bonds that connect the backbone to the nitrogenous bases in the interior are not entirely across each other, but are projected at a specific angle. This causes the sugar phosphate backbone of the structure to be unequally distributed along the helix. Due to this uneven distribution or spacing, different size grooves are formed and are referred to as minor or major grooves, depending on the spacing. So, this is a much closer look at the helix. The major groove, circled in green, will be wider, and the minor groove, circled in purple, will be narrower. This is all very interesting, but why is it important? Well, if you recall the central dogma explained at the beginning, we talked about transcription briefly. During transcription, there are certain proteins that bind to DNA. These proteins are called transcription factors, and they are necessary for transcription initiation. Transcription factors are sequence specific, which means they bind to a specific combination of nucleotides. These factors are able to distinguish between different base pairs and read DNA in a sequence specific manner. How do we connect these back to the DNA grooves? Well, the major groove is the location where the transcription factors bind to DNA. These factors are very important because they can either activate or repress transcription, and as we saw earlier in the video, transcription is a key step in the way genetic information flows. Thanks for watching!